Welcome. I'm going to introduce the topic of de-extinction, so let's get going. Just a note on my fancy headgear, just in case you're wondering. I have recorded this lecture multiple times. One of the things I found out is that the built-in mic in my laptop is just junk, and so it cuts out and so forth. So this is the attempt to try to get things to work so you can hear me. Okay, so I'm going to shrink myself and kind of hopefully disappear here as I go through this. De-extinction. First of all, one of the things that we are going to come across as we go through this material is that one way of thinking about de-extinction is the concept of cloning. Now, it's a little bit more complicated than that, but I just want to be really clear that when we think of cloning, what are we talking about here? Because there's a lot of popular misunderstanding. And for example, if we were to think of what would be a metaphor for cloning, what would describe what cloning is? Cloning is like X, Y, or Z. Um, I think a lot of us would say, well, cloning is kind of like an identical twin. It's like a carbon copy. It's like a Xerox copy. Um, but some might say, well, it's a little bit, different. It's unique. It's like snowflakes. You'll never find two that are identical. Well, let me just clarify that when we talk about a clone, we're talking about, yes, the same genome, the same genetic material, but it's not a carbon copy for the simple reason that its environment, its developmental environment is going to be a little bit different, maybe a lot different. And we know that development is the interaction between the genetic blueprint and the environment in which that development occurs. And so if you think about identical twins, yeah, they may look, identical, but they're not. They're slightly different in personality and in other aspects. And that's because their environment is not exactly the same. And so the same thing when we think about a clone. We're going to clone a species, we're going to clone an individual animal. It's not going to be exactly the same animal. Okay, it's going to have the same genetic material, but the relationship is going to be more like a monozygotic or identical twin. Okay, so we're going to be looking at the terms frozen zoo, de-extinction, of course, somatic cell nuclear transfer, and induced pluripotent stem cells. And these are terms that maybe maybe you're familiar with all of them. Maybe you're not familiar with some of them. And so let's start and see what we've got here. Now, as we before we actually kind of introduce this, let me just also mention that the journal article readings are going to continue as per normal. I've shifted the due dates because we lost a week, and that went out in the updated revised syllabus, and you'll also see it reflected in the dates on the Canvas assignments. If you ever see something that looks goofy, it's probably my mistake. Just let me know. But yeah, so this is due next week. Induced pluripotent stem cells from highly endangered species, published in Nature Methods, super prestigious journal. It's got all the big hitters here in this whole area of kind of assisted reproductive technology. Jeannie Loring from Scripps, Oliver Ryder from the San Diego Zoo, and a postdoc from Israel named Inbar Ben Nun is the lead author on this. So I think you'll enjoy it. Let me just make a little bit of a disclaimer. This is a bit technical, so I have it included in the materials. In that assignment, a glossary and a supplemental reading as well if you're interested. The glossary will help you to understand what all these weird terms are that they're using in this. Once you can understand what some of the terminology is, it's not that difficult conceptually. And don't worry about all the fine details, but you need to know kind of conceptually what they're doing. Okay, so let's move on. De-extinction is really interesting because it was a hot item in the press maybe 5-6 years ago. Haven't heard about it that much now, except now with the northern white rhino and the San Diego Zoo and Scripps working to try to essentially find a way to revive that species. I mean, that species, northern white rhino, is functionally extinct because there's only two females left of that species, and everyone knows that with sexually reproducing species, you've got to have a male and a female. So that's not going to work unless we can find a way to essentially produce northern white rhinos through, again, assisted reproductive technology. So de-extinction is actually in the news again because of that. It's also known as synthetic biology, resurrection biology, the idea that we're going to synthesize life, not exactly the case, or resurrect an extinct species. That's a good Judeo-Christian concept. It certainly is a topic that has excited people a great deal. There's even a book called Resurrection Science. The Wildlife Conservation Society put out a booklet, How Will Synthetic Biology or de-extinction and conservation shape the future of nature? So this idea that maybe this is going to be a game changer. A few years ago, there was a TEDx de-extinction in Washington, D.C., and all these famous, hard-hitting scientists, top in their field, came and talked about it. The link to that, to all of those TEDx talks from that TEDx event are in the module, and you can go and you can watch as many as you want. I'm going to have you view one by Bob Lanza, but you can go and look at others, and they're really pretty cool. So finally, just to not to understate the potential importance of de-extinction, if we think about this stool, and this is a three-legged stool, and the stool is basically the future of species diversity. So you might call it, conservation biology. How successful are we going to be at actually conserving species? And it rests on three legs: protect, conserve, and restore. Protect the species and the ecosystem, conserve it, and restore it. And what some people are proposing is that, well, maybe there's a fourth leg we can add to that. So in other words, if protect, conserve, restore doesn't work, in some cases, we might be able to actually revive or bring back an extinct species. So that's a very exciting concept. It's a game changer in terms of conservation thinking. As you'll see in the last module on Thursday, it also brings up a whole bunch of logistical and ethical questions about whether we should even be thinking about doing this, but we'll get to that. All right, so what is de-extinction? One definition that we might give is de-extinction is the process by which we, human beings, genetically engineered embryos of extinct species, so living species, and hopefully living species that are not endangered, so that we can actually mess around with their embryos. But we genetically engineer the embryos of extant living species to bring back extinct relatives. And so there's a couple things in here. One is we're going to be dealing with embryos, and the other is that we really do need to have a closely related living species to do this. And I'll say this again later, but what this means is that if your hope and dream is to bring back T-Rex or some long extinct species like that, sorry, that's hate to smash your dream there, but that's not likely to happen. Okay, when we think about this whole idea of bringing back an extinct species, I think for many of us, we think about, the popular idea is Jurassic Park, of course, in which, 1993 movie, very popular, but not exactly that way. And it did seem feasible, based on a very carefully researched book by Michael Crichton, very good science writer, sci-fi writer. And what I want you to do now is take a look at a clip of that in which they kind of explain the whole de-extinction process, although they didn't call it that in the movie, and then we'll come back and we'll talk about that. Okay, so let's take a look at Jurassic Park in the next item in that module.